Hello everyone, myself Ritu, NEET Botany faculty. I have strong experience of 15 plus years of teaching NEET Botany and Biology at both the national and international level. My qualifications are MSc Net B.Ed. I have recently qualified IELTS Academic and General with Bench 7 and I am comfortable with various curriculums such as CBSE, IGCSE and IB curriculum. So let's start. Now today we are going to discuss about the dihybrid cross ratio, how we can calculate the dihybrid cross ratio. First of all, we need to understand what exactly is the dihybrid cross. When I say dihybrid cross, you all are familiar that in case of the dihybrid cross, it is the cross involving two pairs of the contrasting characteristics. Like whenever the two pairs of contrasting characteristics are used in the cross, we term it as the dihybrid cross. Let's say suppose here, if we take two characters, that is the height of the plant, as per Mendelian crosses, height of the plant, we know that here the dominant trait, which is represented by the capital letter, is tall, and its notation is capital T, and the recessive trait, that is the dwarfness, it is represented by the small t. Similarly, the color of the flower, if we talk about the color of the flower, then the dominant trait for the color of the flower is red, and it is represented by capital R, and the recessive trait is white, which is represented by the small r. Now, while doing the cross, we are going to take one of the parent as dominant and another as recessive. So suppose if we take the male parent as the dominant, then capital T, capital R representing tall and red. And this is going to be the parental generation, and small t, smaller, that is dwarf and white. Now, during the time of gametogenesis, the gametes found would be capital T, capital R, capital T, capital R, and here it will be small t, smaller, and small t, smaller. As here, the dominant trait is represented by tall and red colored flowers, and here it is represented by the dwarf and white colored flowers. Now, during the F1 generation, we will get, because in the F1 generation, always only the dominant trait is expressed, then it is capital T, capital R, small r. So all the offsprings which are obtained in the F1 generation are tall with the red color. Now, in the next step, we are going to calculate the F2 generation. Like F1 is known as the first filial generation, where all the offsprings obtained with the dominant trait, that is tall with the red color. In the F2 generation, when F1 is crossed with the F1, in that case, when crossing of the F1 and F1, that is capital T, capital R, small r, if it is crossed with capital T, capital R, small r, now here again, this is the parental generation. During the time of gametogenesis, 4 types of gametes are obtained, and this we can obtain by the fork line method. As you can see, capital T, capital R, capital T, small t, capital R, and small t, small r. Again, here it is capital T, capital R, small r, then small t, capital R, small r. So here again, it is going to be capital T, capital R, capital T, small t, capital R, and small t, smaller. Now here, in order to calculate the dihybrid cross ratio, we need to draw a Punnett square, which was developed by the scientist Punnett, and it is known as the Punnett square. With the help of the Punnett square, we will be able to get our required dihybrid cross ratio, capital T, capital R, then capital T, small t, capital R, and small t, smaller. Like this, when we are going to calculate by using the Punnett square, we will get the offsprings. As you can see, there are total 16 squares. And by using this, we can prepare, that is capital T, capital R, then again capital T, capital R, smaller, capital T, small t. Like this, we can fill all the blocks of the Punnett square one by one. And while filling the blocks, we will be able to recognize like what all are the offsprings. Total 16 offsprings will be obtained with the help of the Punnett square, as you can see, and one by one we can fill all the squares. And after filling the squares, as we have reached towards the last line of this Punnett square, you can easily make out by filling them that we are going to get 9 is to 3 is to 3 is to 1. Now the question arises like these 9 are tall and red. As I have drawn the triangle, 3, I'm writing 1, 2, 3. 3 offsprings are tall and white. Again, the 3 offsprings are dwarf and red. And one of the offspring is recessive, which is dwarf and white. So like this, we can say that the phenotypic ratio for the dihybrid cross, by taking the characteristics as the height of the plant and color of the flower, is 9 is to 3 is to 3 is to 1. And the genotypic ratio, as you all are familiar, is related to the genotype. The genotypic ratio is 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1. So like this, the genotypic ratio and the phenotypic ratio can be found out. And for the gametogenesis also, there is a formula 2 raised to the power of n. Since there are 2 pairs of contrasting characteristics, so total, if I'm going to put 2, so 2 square gives 4 gametes, as you can see. So for the F2 ratio, the phenotypic ratio is 9 is to 3 is to 3 is to 1. If more deeply, we will calculate, we will get this like 1 pure tall and pure red. In the genotypic ratio, 1 is pure tall and pure red. If we are going to analyze, that is the first square. Now 2, as you can see, is the pure tall and hybrid red. Pure tall means capital T, capital T. Hybrid red means capital R and the small r. Then again 2, if we are going to see, it is going to be the hybrid tall, that is capital T and small t, and pure red. Even we can write the genotype like 1, we are getting this. Second, we are getting capital T, capital E, capital R, small r. Now hybrid tall, capital T, small t, pure red, capital R, capital R. 4, we are going to get both hybrid tall and hybrid red. When I am saying hybrid tall and hybrid red, that means it is capital T, small t, capital R, small r. Then again, it is 1 is to 2. So it is like capital T, small r, small r. So we can say that it is pure tall and pure dwarf, a pure white. Now, after that, if we take the ratio of 2, then it comes out to be the hybrid tall and pure white. That means this one is capital T, smaller, smaller. Hybrid tall is capital T, small t, smaller, smaller. Then again, this one, it comes out to be small t, capital R. That means the pure dwarf and pure red. So this is small t, capital R. Then the remaining 2 is going to be the pure dwarf and hybrid red. That means small t, capital R, smaller. And the last one is going to be the pure dwarf and pure white. So it comes out to be small t, smaller, smaller. I hope both the genotypic and the phenotypic ratios must be clear to you with the help of this Punnett Square. Thank you so much.